this video we will discuss about the role of Smad3, the proliferation and migrations that is induced by TGF-beta. So TGF-beta has been known to promote EMT in the cancer cells. So here they wanted to know the cellular mechanisms that helps in this EMT behavior and whether this TGF-beta will cause proliferations and migrations, both affect proliferation and migrations in a similar manner or in a dissimilar manner. So in order to understand this impact and why this Smad3 comes into the play is because in the previous paper they have shown that TGF-beta is known to regulate these Smad3 pathways, and we will discuss in this in these results and also in the subsequent videos the role of Smad3 in detail. Role of Smad3 in proliferations and migrations that is induced by TGF-beta. So for now, this Smad3 comes from their previous studies that TGF-beta regulates this Smad3 pathways. So they wanted to check whether this Smad3 plays a role in the proliferations and migrations or not. So first, since A549 Smad3 knockout cell lines, and in the panel A they have shown that these knockout cell lines only affect the Smad3 and the Smad3 phosphorylations, but not the Smad2 and Smad2 phosphorylations. That can be seen here that this phosphorylated Smad3 in the A549 knockdown cells does not shows Smad3 phosphorylations, but when this TGF-beta is induced, it will show Smad2 phosphorylation, but Smad3 phosphorylation is not there. And this for control they use alpha tubulin, and they did their studies Western blot using two different conditions, that is TGF-beta induced for 1 hour in the samples, and for the control they do not induce TGF-beta. And they can see in the normal cell lines there is a that is without any Smad3 knockdown, they see both the phosphorylations when the TGF-beta is induced, and similar results they see in that is similar, I mean for the phosphorylation of the Smad2 results they have seen even in the Smad3 knockdown cells, but the phosphorylation of Smad3 is not there, indicating that Smad3 knockdown only impact the Smad3 phosphorylations pathway, not the Smad2 phosphorylation pathway. Thereafter they check the effect of the cell morphology of this TGF-beta induced after TGF-beta is induced in these different cell lines or different knockout cell lines. First one is the wild type cell lines. Here they see that when the TGF-beta is induced, they see a change in the morphology, and when they induce this TGF-beta in the Smad3 knockout cell lines, they do not see any change in the morphology. But when they again introduce Smad3 using the lentivirus in these knockout cell lines, they can see the change in the morphology again, so indicating that this Smad3 knockout somehow plays a role towards the change in the morphology. And thereafter they check the E-cadherin markers also, and the E-cadherins are epithelial markers, and the E-cadherin is the one of the epithelial markers, and they check the epithelial E-cadherin levels using Western blot, and they are able to see that in the case of control they see a decrease in this E-cadherin levels, indicating that TGF-beta promotes more mesophagous kind of property or suppress these epithelial properties. And for the Smad3 knockdown, they do not see any changes in the E-cadherin levels. But when this Smad3 has been infected in this knockout cell lines using lentivirus, they can see a decrease in the E-cadherin levels, indicating that it's the Smad3 pathways that plays a role towards suppression of E-cadherin signals when the TGF-beta is induced. And the immunofluorescence images also shows the similar results, that is the E-cadherin signals goes down in the normal cell lines after TGF-beta is induced, and for this Smad3 knockout pathways you do not see any change in the E-cadherin signals, but for the when the Smad3 has been again infected using the lentivirus, they see a decrease in the E-cadherin signals. And against this is the immunofluorescence images, and next one is the impact of the cell lines after 24 hours of TGF-β inductions, and we can see the change in the morphology is clear when the Smad3 is either infected or present in the cell lines compared to the case where Smad3 is knocked down. There is not much changes in the morphology of the case where TGF-β is induced or not induced in the Smad3 knockout cell lines. And thereafter they check the migrations properties and the proliferations properties, and panel F shows the bright field images of the migration and proliferation properties when the TGF-β is not induced and the TGF-β is induced after 12 hours. And they did it for all the three conditions, that is for the wild type, and when the Smad3 is knocked down, and when the Smad3 is again infected using lentivirus. And here in the case of migrations, we can see that migrations does not increase when the Smad3 is not present, but when the Smad3 is again infected using the lentivirus or it is present in the wild type, we can see that migrations is significantly higher compared to the case where TGF-β is not induced. But when they check the proliferation results, they see a reverse. They see a reverse observations compared to the migration. That is, the TGF-β is induced, they see a decrease in the proliferation rate. They see a decrease in the proliferation rate, and for the case where Smad3s are present or Smad3 is again induced. But when the Smad3 is been knocked down, they do not see a change in the change or decrease in the proliferation rate. So this indicates that Smad3 plays a role towards upregulation of the upregulation of the migrations and downregulations of the proliferations when the TGF-β is induced. And then they check the wound heal assay, and wound heal assay further substantiate their previous finding that when the TGF-β is present, they see wound recovery is high in after 24 hours compared to the case where Smad3 is knocked down. And when the Smad3 is again induced, they see a recovery in the wound is again higher. And these things are quantified on the right hand side of the panel G, where we can see that the wound recovery is almost same when the Smad3 is knocked down, whether TGF-β is induced or not. But when in the other case where Smad3s are present, they can see a increase in the wound healing recovery. The distance traveled by cells is high in both the cases. This all indicates that the Smad3 plays a role towards proliferations and migrations. That is, its Smad3 has a direct impact to the proliferations, direct impact to the migrations, but a negative impact on the proliferations when the when the cells are exposed to TGF-β. So I hope you understood this basic understanding or the results that they have presented here to show the role of Smad3 in the proliferations and migrations when the cells are exposed to TGF-β. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.